I'm Maria Puglisi, and this is our microbiome authentic research. The purpose of this was to analyze the microbiome of German Shepherds of different age ranges and compare the similarities and differences of the overall gut microbiome. So what is a microbiome? It is the types and the proportion of microorganisms living in a given location. And the gut microbiome is in an animal's gut, and it has been shown to correlate to diet, environment, and several aspects of health. And some of it, the relationship between the gut microbiome and the diet. So diet plays a significant role in shaping the microbiome, with experiments showing that dietary alterations can include large temporary microbiome shifts. So these are my two sources that I found that shows some studies for that. So the research conducted, it was to test intestinal microbiomes and how diet affects it. So human subjects were fed high levels of glucose, fructose, and sucrose in the form of date fruits. So it was found that those who were fed this had increased relative abundance of bifactoroid bacteria with reduced bacteroides. In a separate study, the addition of lactose to the diet replicated the same bacterial shifts while also decreasing the Clostridia species. So this relationship is causal because the change in diet caused the effect on the gut microbiome. So another relationship between the gut microbiome is with disease and health. And so a study that I found is with the immune system must learn to tolerate the commensal microbiota and respond appropriately to pathogens. And in turn, the microbiota is integral to educating the immune system to function properly. So these are the two sources that I found, and the research describes how members of the microbiome community promotes the differentiation of anti-inflammatory regulatory T cells to illustrate how the microbiota can influence immune homeostasis. So this relationship is correlated because they're only relating two variables to come up with a conclusion. So these are just some of the sources that I found for that. And so for this project, the question and hypothesis that we had, the question is, what is the difference in the gut microbiome in different ages of German Shepherds? And the hypothesis is the oldest German Shepherd will have the most diverse microbiome, while the youngest has the least diverse microbiome. Okay, so the animals that we are comparing for this, we had samples number 406, 365, 521, and 643. The breeds, they were all German Shepherds. The 406 was a year and 5 months, 365 was 10 months, 521 was a year and 4 months, and 643 is 2 years and 6 months. So 643 is our oldest, and 365 is our youngest. They're all male except 406 is female. They all relatively weigh around the same weight size. We got 70, 80, 67, 80. They all have average health, nothing's wrong with them. These two take supplements, and they all get regular exercise. So our brief introduction to protocol is and this is a very brief overview. I'm going to go into more detail about what each step does in just a moment, but the brief introduction is we isolated DNA from the stool, we quantified that DNA, then we amplified the variable region of the 16S ribosomal RNA gene by PCR. We checked the products of amplification PCR using aerosol gel electrophoresis. We removed the unused primers, which is like the DNATPS, and the other PCR regimens using commercial PCR cleanup kit. And we added a unique set of sequence tags to the end of the amplicon from each sample in an indexing PCR reaction. We cleaned up the indexed PCR reaction product and quantified the amplified DNA and prepared a dilution of our amplified DNA so that it was ready for sequencing. So finally at the end we used it was the Illumina Corp's MASEQ, and it was used, which is an automated DNA sequencer to determine the nucleotide sequence of the various amplicons in the samples. So now to go into more detail about that, our materials and methods, we had the purification of DNA from the fecal matter. That was performed with the QIAGEN kit. So we followed the manufacturer's instructions as described in the lab manual, so no intentional or accidental changes were made. The concentration of DNA after purification was done with the Zymo kit. So we followed the manufacturer's instructions again as described in the lab manual, so no intentional or accidental changes were made. And so during the Zymo kit, the DNA purified from the first steps were bound to a filter column and then rinsed of impurities and then eluted into a special buffer. So this concentration, we concentrated it at tenfold. And next we quantified the quantification of DNA in these samples. So we used absorption spectrophotometry with the UV spectrophotometer, which is the nanodrop. So this spectrophotometer produces ultraviolet light, and DNA absorbs light very well at 260 nanometers, which is in the UV range. So that means we can use the absorption at 260 nanometers to calculate the concentration of DNA in our solution. Proteins absorb light at 280 nanometers, so we can use the ratio of absorbance at 260 nanometers over 280 nanometers as a measure of purity of our DNA. Okay, so next is the amplicon PCR, which is the polymerase chain reaction. It amplifies or copies small segments of DNA. So we amplified the 16S ribosomal RNA gene, specifically V3 and the V4 region. We wanted to analyze the prokaryotic 16S ribosomal RNA gene because this gene contains 9 variable regions interspersed between conserved regions. So it is run for 25 cycles, and the target DNA is first denatured by heating to 95 degrees Celsius. The sample is then allowed to cool so that the primers combine by base complementation. Once the primers have bound, the temperature is raised to 72 degrees Celsius so that the DNA polymerase combined and start synthesizing the DNA. So in order to change reactions temperature quickly, the PCR is conducted in the thermocycler, which is programmed to change the reaction temperature quickly. So that's how we did the amplicon PCR. Next we had gel electrophoresis. Electrophoresis is a technique on the ability of charged particles in solution to migrate in an electrical field. So we separate DNA fragments of different sizes using the aerosol gels. So the voltage we used, it was 250 volts. So the distance a DNA molecule migrates is inversely proportional to the logarithm of its molecular weight. So knowing this relationship, it allows us to determine the size of the DNA fragments that we are studying. So after we did gel electrophoresis, the next thing we did was PCR cleanup. We used the aging core, it was the Ampere XP kit, to clean up the amplicon from our amplification PCR action. We followed the manufacturer's instructions, so no accidental or intentional changes were made. The cleanup removed, it was the components of the amplification PCR buffer, so these are the things that it cleaned up, as well as the unused and forward and reverse primers. So this is important, and it's very critical to the success of the next indexing round of PCR, especially if the removal of the unused old primers, because if they were to remain in the solution with the amplicon, they would interfere with the indexing PCR by binding to the indexing PCR primers. So I'm going to talk about that now. Indexing PCR it's essentially we're adding tags to them, but we added a unique set of short sequence tags to the end of amplicons from each sample via an indexing PCR reaction. So when we sequence the DNA, we will know which sequence came from which sample via those tags. We use 2 primers, like with the amplicon PCR, but in this case, only the 3' portion of each primer attached to the amplicon. The 5' end of each primer contained the unique sequence tags needed to identify the sample it came from. So because we are only trying to add these unique pairs of sequence tags rather than make large amounts of new amplicon, our PCR reaction only ran for 8 cycles as opposed to the 25 cycles used in the amplification of the PCR. So next we have the PCR cleanup, and that's basically the same as the first one. We used the same kit and everything, pretty much the same procedures. So after we do another PCR cleanup, we did the quantification of DNA indexing PCR products, which is the fluorometric DNA quantification. We used the fluorometer. Fluorometric assays are more accurate than the spectrophotomic assays because they are based on the shift in fluorescence properties of a molecule after binding to the target. So UV absorbance measurements are not selective and cannot distinguish DNA, RNA, or protein, so values are easily affected by other contaminants like these. So, and variations in base composition. Also, the sensitivity of spectrophotometry is often inadequate, so it prohibits the quantification of DNA and RNA at low concentration. So that's why we used this one instead of the spectrophotometer. And after that, we did our DNA sequencing, which is the nucleotide sequence of each indexing PCR fragment. We determined it using the MASEQ next generation DNA analyzer. So for this experiment that we were doing, our independent variable was the age of the German Shepherds. The dependent is the variability of bacteria found. And then we had our negative and positive controls, and I go into more detail about them down here. Positive control that we used, it was purified DNA from a pure culture of E. coli, which is one of the many species of bacteria that live in the human gut. The reason we use a positive control is for the possibility that none of the other reactions produce a product. So if we run those reactions without positive control and not see any product, we would not know if the lack of product was due to a problem with the extracted DNA or some defect in our protocol or reagents. If we include a positive control and see product from the positive control, but no product from the other samples, we can safely say that the lack of product is due to some problem with the DNA isolated from scat samples. And then our negative control, we had no template, so no isolated DNA was added. So that is, we replaced the amount of purified DNA with water, and in this reaction we are testing for cross-contamination from other sources. So no products should be seen from this reaction in negative control. So next I'll show you the results of all this. Okay, so here's the results from what we tested, and the initial UV spectrophotometry, which is the DNA quantification results. We had samples, here's our samples, and this is the 260-280 ratio. We had 1.89, 1.25, 1.9, and 2.02. And so these are our results from that. And then we had the gel electrophoresis results. This is our negative control, positive control, 365, 521, 406, 643. So the size of the amplicon is approximately 550 base pairs for each sample. Our positive control is at 550 base pairs, and our negative control has produced no results, which is what we wanted. And house sample 365 produced no results. So we had to use data from another group. See here, it didn't produce a result. So the quantification of cleanup amplicon PCR reactions, we were about 550 base pairs for all of them except for the negative control and the sample number 365, like I said. That was the only unsuccessful read. And then quantification of indexing PCR products. This was our phelomic quantification. Here's all the results for that right here. And so to get into what we had sample 406 when we got our results back. For the kingdom levels, we had, it's mostly bacteria, it's 99.98% bacteria, and the other is 0.02%. And that's for sample 406. And then for the order classification results for 406, the top one that was present was the clostridiols right here, and then we had the bacteroidales right here. Those are the top two results, and these are the other ones that are present for the order of classification result. And then also for sample 406, so for the genus classification for sample 406, Praevotella was the top one, and then Megamonas was the other one, and here's all the other results from that. And sample 365, the top kingdom, the kingdom classification results, again, it was mostly bacteria, which is what we want, 99.97%, and then 0.03% is the other. And sample 365 for the order, the top one was the bacteroidals and the clostridiols, and then here's the rest of those results. Also for sample 365, for the genus classification, again, it was, we had Bacteroides and we had the Praevotella, and here's all the other results for that one. Okay, so for sample 521, for the kingdom, we had bacteria again was the most prevalent. It was 99.96%, and other was 0.04%. And also for sample 521, the order classification results, we had the bacteroidals and the clostridiols, and here's the rest of that sample. All the other results for that sample. And then again for sample 521, the top genus classification results, it was the Bacteroides and the Praevotella, and here's the rest of those results. And for our last sample in 643, the kingdom, we had mostly bacteria again, which was 99.97%. Other was 0.03%. For the order classification results, the top one was the Clostridiols and Bacteroidals, and here's the rest of those, and there's the pie chart. And again, for sample 643, the top genus classification results, we had the top one was Prevotella, and then second was the Megamonas, and here's the rest of those. So these were our overall results for that. So the categories of other essentially means that it could have been some other contaminant or some other thing besides bacteria that was in the gut that we examined, and unclassified means that it was bacteria that they just couldn't figure out what type it is. Okay, so for our discussion, DNA extraction from scat was run through the UV spectrophotometer after being concentrated. The ratio of 260 nanometers over 280 nanometers, which it should be between 1.5 to 2.2, it gives a measure of the DNA's purity. So readings below 1.5 indicates possible contamination, and all samples passed the 1.5 mark except for sample 521 for us. So not having pure DNA may interfere with the accuracy of further results. The electrophoresis reading showed no DNA in sample 365, meaning not enough DNA was detected for sequencing. So the negative control also contained no reading for DNA, which indicated that there was no cross-contamination. So every other sample contained DNA. We used the ratio absorbance of 260 nanometers over 280 nanometers as a measure of the purity of our DNA, like I said over here. But pure DNA has 260 over 280 ratio, which is approximately 1.5 to 2.2. So ratios below 1.5 indicate possible contamination with proteins. If the 260 over 280 ratio is far from acceptable range, it means that our estimate of the DNA concentration may be inaccurate, and the contaminating protein may interfere with some of the other steps that we did. So the sample numbers that we had, the readings that we had, so as long as the samples had over 1,000 reads, then it's adequate to use, and all of our samples had over 1,000 reads. We had 13,164, 14,301, we had 251,242, 10,633. So they were all adequate to use. So now, one of the types of the bacteria at the genus level that I wanted to talk about was the Bacteroides. So Bacteroides are characterized by their mutualistic behavior, and they are typically present in the gastrointestinal tract of mammals to function as normal flora. So Bacteroides, they're capable of breaking down and processing large complex molecules within the intestine. So the Bacteroides are able to benefit the host by preventing infection by potential pathogens that may colonize and infect the gut as well. So due to the abundancy of the Bacteroides within the gastrointestinal system, Bacteroides constitute a significant portion of the fecal bacterial population. So these are the two sources that I found to talk about this, and Bacteroides were present in some of our samples that we analyzed. So in the end, addressing our question and hypothesis, I cannot say that our data concludes our hypothesis to be correct, because the data from the oldest German Shepherd, which was sample 643, it was quite similar to the data from the youngest, which was 365. So a lot of factors contribute to this that make it different, because not all of our samples were male. Sample number 406 was female. So we can't exactly deny or accept our hypothesis, because there are too many factors that can contribute to it being different, besides the fact that there is an age difference. So these are our results, and that is the end of this.